Replication. So synthesis of daughter DNA by parent DNA is called as replication. So from a parent DNA, from one DNA, two daughter DNAs are synthesized. So that process we call it as replication. Right? So whatever the information in the parent DNA is, that is transferred to both the daughter DNAs. So one of the parent strands is seen in both daughter DNAs. So such type of replication we call it as semi-conservative replication. So that's how we can define replication. When you see in the picture, we have a parent DNA. From that, each DNA acts as a template for the synthesis of new DNAs. So two new DNAs are synthesized from a single DNA. So whatever the information in the DNA is, that is copied into the both the DNAs. So this is called as replication. And coming to what are the materials required for replication? So we require a DNA template, we require deoxynucleoside triphosphates, and we require DNA polymerase. So apart from that, we require some other enzymes and proteins like we require helicase, we require primase, and we require single strand binding proteins, we require DNA ligase. Like that, we require all these topoisomerases. We require these other enzymes and proteins are required for the replication process. So all of them are the materials required for replication. And coming to mechanism of replication. So this can be discussed under three headings like you can see it initiation, elongation, and termination. So these three will occur in case of these are the three phases of replication. So initiation, elongation, and termination. First, we'll discuss about initiation. What happens in initiation? So in case of initiation of replication, first, so as I told you, we have some proteins, other proteins. So one of them is a protein called as DNA protein. So that DNA protein in case of prokaryotes, the function of that DNA protein is so that binds to a specific region on the DNA, we call it as DNA boxes. Right? So that DNA boxes is present in the ORI region. So in order to the initiate replication, first region of so we have to that enzyme has to recognize a particular region on the DNA. We call it as ORI site, origin of replication, has to be recognized to initiate replication. Right? So that origin of replication has some specific sequences, and in that origin of replication we have DNA boxes. So those DNA boxes are identified by a protein called as DNA protein. So that DNA protein binds to those DNA boxes, and when it binds, it melts the DNAs. So it melts that DNA, and melting is whenever that melting occurs, the two strands of DNA are separated, and that is brought by DNA protein. So here the important point is in order to initiate the replication, we have to identify the origin of replication. So that origin of replication has some sequences, and those sequences are to those sequences a particular protein binds, that is called as DNA protein. Right? So once it binds, it leads to separation of DNA strands. Right? So that's how origin of replication is found in case of initiation of replication. So coming to what happens when that DNA protein binds to that DNA boxes in the origin of replication. As I told you, it melts the DNA strands, separates the DNA strands. So when DNA strands are separated, it results in the formation of replication fork. Right? Replication bubble is formed, and that leads to formation of replication fork. Right? So once the two strands are separated, next the enzyme which comes into action is helicase. The function of helicase is so that helicase separates the two strands. So and for its separation requires the energy, and that is supplied by ATP. So using the ATP, a helicase separates the two strands of the DNA. Right? So once the helicase starts separating the two strands of the DNA, and to prevent the rebinding of separated DNA strands, we require the help of another proteins called as single strand binding proteins. So these single strand binding proteins, they bind with each single strand. They won't bind with the double stranded DNA, but they bind with single strands of the DNA. And once they bind with the single strands of DNA, they prevent the rebinding of DNA. Whatever those are separated by helicases, their rebinding is prevented by single strand binding proteins. So that is the use of SSB proteins. So by this way, so helicase starts separating the two strands of the DNA, and single strand binding proteins, they bind with each single strand, and that forms a replication fork. Right? So that's how replication fork is formed. And coming to elongation of DNA strands. Here, so when you see, so when a replication fork is formed, the DNA is synthesized in two ways. In case of one strand, DNA is synthesized in continuous manner, we call it as leading strand. In another strand, it is synthesized in fragments, we call it as lagging strand. So DNA synthesis occurs in both strands after its separation. So both strands of this DNA, they act as template for synthesis of new DNA. But in one strand, the synthesis occurs continuously. We call it as continuous strand, a leading strand. Other one is discontinuous strand, a lagging strand. So in both the cases, the synthesis occurs in the 5' to 3' direction. So templates are read in 3' to 5' direction, but synthesis of strands occurs in 5' to 3' direction. So whether it is leading strand or lagging strand, both the strands are synthesized in 5' to 3' direction. Right? So in order to synthesize new strands, we require RNA primer. Right? So this RNA primer we can compare with that of glycogen synthesis. In case of glycogen synthesis, we require a primer in order to add the incoming glucose units. Right? The same way in case of replication also, for the synthesis of new strands, where there is addition of incoming nucleotides is there, deoxynucleotides will be added by the action of DNA polymerase 3. So for the action of the DNA polymerase 3 to polymerize the deoxynucleotides, incoming nucleotides, we require a RNA primer. So that RNA primer is synthesized by an enzyme called as primase. So primase synthesizes the RNA primer, that is a short segment of nucleotides. And once RNA primer is synthesized, and now the incoming deoxynucleotides are polymerized by the action of enzyme called as DNA polymerase 3. Right? So that happens in case of both the strands, we require RNA primer. So when you compare, in case of leading strand, we require only one RNA primer, whereas in case of lagging strand, we require multiples of many RNA primers. So that is the difference between requirement of RNA primers in case of leading strand and lagging strand. Right? So there is continuous synthesis of synthesis of DNA in case of leading strand with a single RNA primer. So when you see in case of lagging strand, we require many RNA primers. So one RNA primer is synthesized by the primase. After that, to that RNA primer, the deoxynucleotides are added by the action of enzyme called as again DNA polymerase 3. Right? And that DNA polymerase 3 can add deoxynucleotides until it comes in contact with the next RNA primer. So there it stops its action. Again, it adds the deoxynucleotides to the other RNA primer. So like this, so RNA primer is synthesized to that deoxynucleotides are added by the action of enzyme DNA polymerase 3 until it comes near to that RNA primer. So once it approaches that RNA primer, then the action of DNA polymerase stops. Then, so sequentially this mechanism occurs until the whole chain is copied. So then what happens in the lagging strand is once all the Okazaki fragments, so we call RNA primer with short segment of DNA as Okazaki fragments or Okazaki pieces. So once all the Okazaki fragments or pieces synthesized, then one more enzyme comes into action, that is DNA polymerase 1. So the function of DNA polymerase 1 is it removes those RNA primers. So RNA primers will be removed by DNA polymerase 1. So once that RNA polymerase are removed, the gap which is created after the removal of RNA primers is filled up again by the action of enzyme DNA polymerase 1. So DNA polymerase 1 removes the RNA primers, and also it adds the deoxynucleotide. So it has polymerase function and also excision function. It removes the RNA primers. So once RNA primers are removed and that gap is filled by the deoxynucleotides by the action of enzyme DNA polymerase 1. So then again we have a small gap between these Okazaki fragments after removal of the RNA primers and synthesis of a short segment of DNA, and that gap is filled again by the action of enzyme called as ligase. Yeah. So RNA primers are removed by DNA polymerase 1, new strand is synthesized, new segment of DNA is synthesized, and after that there is some gap, and that gap is again filled up by, that is joined by an enzyme called as ligase. So ligation requires energy, and that is supplied by ATP. Right. So this is how synthesis of DNA occurs in the lagging strand. So for this we require many RNA primers, action of DNA polymerase 3. After that DNA polymerase 1 is required, and also we require the help of DNA ligase. So this is how the synthesis of DNA occurs in both leading strand and lagging strand. Right? And this is what happens in case of elongation. And coming to termination, there is particular sequence in the DNA where a protein called as TUS protein binds, that is called as terminus utilization substance. So that binds to the TUS protein binds to the particular sequence on the DNA, and that leads to termination of replication. So this is how initiation, elongation and termination of replication occurs. So once termination occurs, so now we have two new daughter DNAs which are synthesized from a single parent DNA. So both of them have a strand which is derived from the parent. Right? So such type of replication we call it as semi-conservative replication. Coming to inhibitors of replication. So there are some antibiotics and anti-cancer agents which act as inhibitors of replications, and those are nalidixic acid, ciprofloxacin, and 5-fluorouracil 6-mercaptopurine. So all of them, they are nothing but they are inhibitors of replication. And when you see nalidixic acid and ciprofloxacin, they are antibiotics, antibiotic agents, antibiotics, antibacterial in action. And when you see 5-fluorouracil and 6-mercaptopurine, these are anti-cancer agents. So they can inhibit replication in our body. And when you see nalidixic acid and ciprofloxacin, they inhibit the DNA replication in bacteria. So these are the important inhibitors of replication. So till now we discussed about the what is replication, what happens in case of semi-conservative replication, what are the stages of, what are the phases of replication, and what are the important inhibitors of